Good afternoon, everyone, and welcome to the first webinar in the Mushroom Farm Hygiene mini-series. The webinar series is facilitated by the Australian Mushroom Growers Association, with workshop presentations delivered by Dr. Warwick Gill of the University of Tasmania and Australian mushroom expert Judy Allan. This webinar is brought to you by the AMGA managed project MU22010 Mushroom International Inbound and Outbound Study Tours, which is designed to facilitate educational opportunities for industry personnel to upskill the broader Australian mushroom industry. It also links with MU21007 Pest and Disease Management for the Australian mushroom industry. Both projects are funded by Hort Innovation using Mushroom Research and Development Levy, contributed by growers and funds from the Australian government. It's great to see we have a mix of farm owners, managers and operators on the webinar today. In fact, we have 82 participants on the call, which is phenomenal. Thank you for joining us and thank you for your patience while we began. And as you will undoubtedly know, good pest and disease control begins and ends with good farm hygiene, a topic which can potentially slip from top of mind when you're busy growing. We may have time for Q&A at the end of today's webinar, so please use the chat function via the platform and we will respond to your answers at the end of the presentation if we have time or at the next webinar. If you have any specific pest and disease or hygiene questions relating to your farm, please reach out to Judy or Wal directly for a confidential conversation. Before we get started, I would like to advise that this webinar is being recorded and will be sent as a follow-up to all registered participants and later will be housed on Agora, the mushroom industry pest and disease website. Visit Agora's website at Agora, that's agora.australianmushrooms.com.au, which is password protected. If you need to be set up with access to the website, please contact Judy Allan directly. Without further ado, I will now hand over to Judy Allan to start the presentation.
Okay. Can you hear me, Warwick?
We can hear you.
Okay, well, welcome. Sorry, that was my technical issues that slowed things down. This is session 1 of a 4-part series. I'm going to do some introductory statements and descriptions about farm hygiene. And then Warwick's going to do the second part and just go into a bit more detail. Next slide, thanks. So when I do this pest and disease work with the industry, I often think about in the 5 moments of need, and I'm the interface of a project with the growers usually, and they come when things go wrong, point 4. But I know in this audience we've got people who are learning for the first time, people who are learning more, and hopefully we're going to let you learn a little bit to apply what you've learned. And of course, when things go wrong, it's knowledge that makes such a difference. So farm hygiene is a system of defence against pests and diseases aimed at reducing the chances of the enemy getting in and minimising the threat when a pest or disease does get in. So they are fighting words or war words: enemy, defence, minimising when they do get in. So next slide. So hygiene is a major component of IPM, Integrated Pest Management. Integrated Pest Management is perhaps a more modern word than hygiene, but we've done farm hygiene in the mushroom industry for many years, whole farm hygiene. And it is the same thing as Integrated Pest Management, but we're going to refer to it more in this series as Integrated Pest Management. So without hygiene in the early stages of the growing cycle, other containment measures used in later stages will not be effective. The objectives of a farm hygiene program are to exclude the pests and diseases from the production cycle, eliminate them, or at least contain them if and when they occur, to minimise losses. So in an intensive situation like mushrooms, it's not really feasible that you'll ever have 0 pests and diseases. That it's more about eliminating them if they come and containing them when they come. And one of the things that's really important in that is the destruction of any pests and diseases present at the end of cropping. That's a lot easier on farms that can cook out, and we know that most block farms, the smaller farms, can't cook out. So other techniques have to be used, and that'll be more talked about in another session. Next slide, thanks. So back to fighting words. Mushroom pests and diseases are survivors. They don't give up without a fight, and they come back if given half a chance, and they do not change their behaviour to suit your convenience. And that's so true. Next slide. So I'm doing the general bit for you now, just that the general hygiene practices or principles are to start off clean, protect the crops from infection with physical exclusion. That means filtration, closing doors, fly screens, things like that, to prevent disease spread within crops, and monitor and manage levels right up to the end of the crop. Don't say, oh, that room's too diseased, I'll close the door and it'll get tossed out on Monday. That's the worst thing you can do for hygiene, because what you're doing is breeding up levels of inoculum that will then got a much better chance to spread around the farm. Next slide, thanks. So principles of good hygiene: kill off the disease at the end of cropping. Cooking out in situ is the most effective. Some farms, like a tray farm, they take their compost out, the trays out of a room and take it to another spot on the farm and cook it out. When it's cooked out in situ where it's growing, it is more effective, because when you open the door and take the older crop out, you can release things around the farm. So, of course, if levels of disease become excessively high, it is desirable to terminate the crop early. And that's all about minimising the amount of flies or inoculum that are there to spread, because the war of hygiene is a numbers game. Next slide. So common sources: where do all these pests and diseases come from? They come from compost raw materials, and they're still in phase 1 compost. Some of the things are in phase 1 compost, but the compost is prepared over 2 phases, and the second phase is when the temperature and ventilation is highly controlled through a series of stages, and that is actually the cleanest stage of the compost. Is not phase 1, but it's at the end of phase 2 when it's spawned. No, you have to go back, Warwick, sorry. A source of pests and disease can be encasing materials, and mostly when we visit farms we see that bags are broken and stored badly. So it's a difficult thing to prove, but it is understood that you can be adding pest and disease encasing materials to your crop. So dust and soil, and there's in another session we're going to talk about farm dust, and because it can vector things around the farm, and natural field populations are those flies that might live out in the environment, mites that live in, well, the compost raw materials, nematodes can be in them, insect pests, things like that. And then a common source of disease is infected mycelium in spores, and that's in particular in relation to virus and mushroom virus X and La France disease. And of course, a source of pest and disease is the old crops, and a hygiene principle is that the older the crop is, the more likely that a pest or disease is present. And another hygiene principle is the earlier the infection occurs, the highest potential for loss happens. So there's a number of principles around all the things that we talk about. Next slide, thanks. So the basis of a hygiene program is that prevention's better than cure. Once established, the pest or disease can cause a direct loss in yield, and that can be from a few percent to 10% to 30% and up to 95, 100, and lock the door, close the farm. And the ones that have the greatest potential for direct losses in yield are the viruses and Trichoderma aggressivum. Whoops. And then there's indirect losses in quality, and also with picking, and the cost of pesticides and containment measures, and that can be quite significant. I've talked to big farms when they've got a bubble problem and they've spending 70 man-hours a week. They were spending 70 man-hours a week containing their disease, doing spot treatment. So that's a significant cost. Next slide, thanks. So this is pretty obvious. On a farm with no pest and disease, happy faces walking around the farm. A farm with problems and pest and disease, sad faces. So I'm just going to show some of the, next slide, some of the things. So on the bed you can see mushrooms, and on the left you can see green mold. And so they're just unsaleable mushrooms. They're nice shape, they'll grow into more yield, but they're unsaleable. They have to be dealt with. So they're a big cost to the farm. Next slide. So nice mushrooms again, but there's red pepper mites on them. They're unsaleable. Red pepper mites are normally a secondary thing. So that's they're millions and millions on top of the mushrooms. They were probably picked, well, they were picked and they weren't present. They came to the top in the cool room. And that photo was taken when mushrooms came out of the cool room. And red pepper mites normally are associated with green mold in the compost, and they will swarm once they run out of food and they're trying to hitch a ride to somewhere else with more food. So some of these pests and diseases can be indicators as well. Next slide. So pickers are usually the eyes of the farm, because a lot of the pests and diseases don't express till during picking. So people early in the cycle do a lot of work and preventative measures, but it's the pickers who really see the result of all that. So those orange little grubs, they're larvae. They're called orange cessids. They make the mushrooms unsaleable too. They're normally associated with peat, but peat storage actually, because they have a life cycle and they spread around with adults, as adults, and then they express on the mushrooms. And you can see that that's a knife handle behind, so they're pretty tiny. So if in bad lighting, people mightn't notice it, but we have to look very closely for some of these things, because that's a characteristic, is that pests and diseases are a lot smaller than the mushroom, and they have a lot quicker life cycle. Next slide. So unsaleable mushrooms can be caused by bacteria as well. A healthy mushroom has, say, 30 million bacteria on its cap, and an unhealthy mushroom might have 100 million. That's the numbers game of bacteria. And over the years, things have changed, and we actually have, we used to only have bacterial blotch caused by Pseudomonas thalassii, which is the one on the left, A, but there are now known other blotch causing organisms, and we knew about ginger eye, but then there's Salmonii. So even within a disease, there can be variants of a disease. Next slide. Thank you. So that's a pretty sad situation. We have very difficult operating climate at present. And a number of growers say to me, well, third flushes, we need a third flush because we have all those expenses and we need that that's when we can start making a little bit of money. So that photo shows a very sad case of spot the healthy mushroom. There's actually no healthy mushroom. That's third flush. So there's a lot of, there's salting, mightn't be correct, but like there's been an attempt at salting. There's greatly reduced yield because to have that much bubble in third flush, you'd have to have some in second, might have been some in first and been watered. And it's a very unhappy face. Next slide, thanks. So with these diseases, you need to be careful. Don't be mistaken seeing one of these photos that we put up and saying, that disease is always going to look like that, because it doesn't. This is leccanicilium, which was called, its more scientific was verticillium, but we mostly refer to it as dry bubble. And these are the range of symptoms that can be expressed, and they express differently according to when the spore lands on the mushroom. But I think that makes training a little bit difficult for pickers, because I see that on the signs on doors when people put good awareness things up. If you see this disease, tell us. But which photo do you choose? So I think I would encourage you to all use the range of symptoms in your training and choose the one that's mostly present in your farm. And like some of these symptoms tell me things. The second last photo, the spotting and branding on the caps, means that a spore's landed on that cap, and that's the reaction of the mushroom. The top left, the distorted pin soon after emergence, that means that the spore landed in that spot when that spore was, that mushroom was very small, and it has the real characteristic bubbles. And the tricky thing is the top right-hand one, that is still dry bubble, but some people see those drops and they think, oh, it's wet bubble. So look closely and, take the corrective action, but sometimes it is advisable to make sure you've got the right diagnosis. Next slide, thanks. So on Agora, so I wrote the address of Agora there for you. It's Agora, australianmushrooms.com.au, no dot between Australia and mushrooms. There are a number of recognition books, and that slide is inserted exactly how I wanted it to. It's actually a pocket book that you can download off Agora, print out, and people can have it. And then instead of it going from room to room, it can be destroyed and not spread spores. So there's 3 pocket books. So next slide, thanks. Warwick and I work in different states, so we don't get to practice this. I'm not very good at computer, so or at Zoom. I'm really pleased that Warwick changes my slides for me, because I don't want to do the uploading at home. So this is another pocket book that we have, and another disease. This is some symptoms of virus, and in the old days, the virus we had was La France disease, and this is a modern disease associated with bulk handling of compost, and there are a number of symptoms, and it evolved over time. So bare patches were traditionally which is the right-hand photo, were traditionally something that identified that a virus was around and you needed to get a test. And you would sample the mushrooms on that right-hand side. You would sample the mushrooms immediately outside the bare patch, because that's likely where the strongest piece of virus would be. However, there's other symptoms, and one was referred to, the next one to the left is brown mushrooms. And that is, people used to think, oh, there's a brown mushroom in amongst the white mushrooms. They must have mixed it up at the spawn lab. But in fact, that is one of the symptoms of mushroom virus X syndrome. and patch disease is another symptom. So just go to the next slide for me, Warwick. On the left-hand side, the patch disease, that's a real characteristic thing, the swirling of the patches along the bed. So we have seen all these symptoms here in Australia. Don't think this is an overseas disease. And we have got a very good diagnostic lab for mushroom virus, and some of the symptoms of mushrooms can be confusing, and the best way to identify if you've got virus is to get it tested in a lab. Yep, that's fine. Thanks, Warwick. So the next disease that causes majors and farms to close, it can start off with a little bit and get spread. Farms in Europe have closed down from it. The thing about it, it's called the aggressivum. It's aggressive. Only a little bit of green mold will spread, and there's a range of different green molds, and the one that identifies this one is testing, but also all the crop loss that goes with it, because the slower, less aggressive ones often just grow in the beds on third flush. So you still have mushrooms, but they're spotted, whereas aggressivum, you get dramatic crop losses, 95% close to 100% closed doors. Hopefully, it's addressed before it gets to that point. And the point is, contaminated equipment and machinery can infect phase 3 compost. So if clean compost comes onto the farm, if there's still contamination around, if there's still spores of this around, it takes very few spores to cause problems. And really, hygiene is so important to control these last two diseases, this aggressivum and mushroom virus X. Next slide, thanks. So this is where we get that. It's the whole farm approach to farm hygiene, because clearly research has indicated that all compost mixing operations are high risk to spread it. So look how much there has to be cleaned on that equipment. It's not really it's a very difficult disease to manage once you have on the farm. Next slide. So while sometimes the original source of disease is debatable, once present on a farm, the main source of spread comes from a diseased room. And I've put this up. It was the moonscape on the left-hand side. I took that photo on a little farm that had cobweb, and they couldn't keep up with things, so they didn't spot treat to the end. They didn't cook out. And I happened to be on the farm the day they tipped out that room, and Warwick had given me some plates. So the first plate, I just exposed a couple of flies on it, and that's what ended up growing. The second plate, I just exposed outside the room that was getting emptied, and that's what grew. So that cobweb species has that distinctive pinkness that goes with its growth. And then look how dirty the owner's sleeve was. So when you haven't got a disease on the farm, you can get away with a lot of things. But as soon as you have a disease on the farm, that war starts, because that's their survival mechanism, is to release their spores and find another home for themselves. Next slide, please. So early detection and spot treatment must be a key focus to contain the disease once it's there. And this aspect of management, so spot treatment and early detection, can differentiate farms that have serious, widespread and significant losses, and those that have disease appearing at times, but it never eventuates to anything major. So a farm that's like that previous slide with all that cobweb, that farm's probably quite in tune with early detection and spot treatment, because they're doing it all the time. But a farm that's clean, then when something comes along, unless people are looking right and do the right thing with that disease that's needed for that disease, it can get out of control. So I'm pleased that there's such good attendance of farms here, because if you're successful at pest and disease, avoiding pest and disease, you've got to keep the defences up all the time. So continual awareness and reminders are very good. Next slide, thanks. So another base characteristic of the enemy is that it has a shorter life cycle than the mushroom crop. So these are the main mushroom flies. The fly on the left is a phorid, and the fly on the right is a scarrid. And each of these individual pests have slightly different biology and behaviour. The phorid, while one phorid can lay 50 eggs, and that's so one phorid 50 on your farm. Just think of it, if it's at filling, in 3 weeks' time, there'll be 100 flies. Sorry, there'll be 50 flies. And if it was a scarrid that was there on filling day, there'll be 100 adult flies there. And then by the time that in 3 weeks' time, there's another generation. So that's what makes these guys so competitive, and it identifies where you really have to focus in very closely to keep the numbers down. So flies, big ticket item is to reduce the number of flies on the farm on the day before filling and the day of filling, and knock them down quickly so they don't lay their eggs and do their generations. Next slide, thanks. I think I'm nearly finished. So in amongst the group, there are different farm production systems. There's trays, there's shelves, there's blocks, and you'll be able to have this video later. So I'm not really going to go through a lot of these things, but I always say mushroom farms are like people's noses. They're all different. So there's a lot of common things there, but there's also some differences according to the individual site and to the system. The biggest factor of block farms, I think, is no cookout at the end. Shelf farms is the doors are open when the compost is getting filled. And on smaller shelf farms, it often takes quite a long time to fill. So a fly has plenty of time, for instance, to get in. And Warwick's going to demonstrate very well how these flies actually spread disease. So the age of the farm makes a difference. And farms don't stay new for very long, and all these things start happening. All farms pick, and picking trolleys move from room to room. So there is a lot of common things, but there are some different things. Next slide. So some events and situations that increase the likelihood of increased pest and disease in a mushroom farm. It could be also called, how do you make a little bit of disease a lot of disease? Well, this first list is about people, actually, and the take-home message that I have is that people make systems work. And some of the things that happen, and in the current operating climate, a lot of these are happening. Staff shortages, staff turnover, new inexperienced staff, lack of training, poor morale. People, when there's pests and disease, you know, it's not a nice. The job. It takes a lot away from the job of picking, and people get disheartened by it. Staff not following farm procedures to farm standard, and key staff on holiday. And believe me, they are often implicated on when disease or pests just blow up, that they were just a few and then there's a lot. Just next slide, thanks. So this is just the non-people side, and one that's really key is not putting enough emphasis on cleaning. And in session 2, we'll be doing a lot about cleaning and disinfecting. Leaving growing rooms doors open when they don't need to be. That's a real pet hate of mine. People do have to go in and out, but you lose all your physical exclusion when you leave growing doors open. Watering onto disease is a very common thing that happens, particularly on weekends, because through the week people seem to be better organised, but on weekends the spot treaters are often not there, and then watering happens, and then the disease spreads in that area of the bed, gets onto the floor, gets into the dust. So watering onto disease and spot treating are really something that needs to be managed closely 7 days a week. So we've seen input changes increase pests and disease on farms. Like once upon a time, you could we'd give a recommendation on what chemical or disinfectant to pesticide or disinfectant to use, and you could go buy it. But these days, you can't assume you can buy anything. So sometimes what's used is different, and it mightn't be the right thing. If they're forage, you use one chemical, and if they're scourge, you use another. And as is, there are better fungicides to use with fungus diseases. Some you choose according to the target. So compost, I've got input changes. Compost arriving to the farm too wet or too dry, it just makes management so much harder and variation. And like if it's too wet, that can favour diseases to grow in the compost. So what we're really about in a mushroom farm is to have conditions that favour the mushroom and good quality. And as soon as they get less than ideal, that these survivors use that as an opportunity to get hold of the crop. So building activities can impact, particularly with bubble, Lechinocilium. When there's excavations around the area of the farm, often bubble then appears on the mushroom farm, and it can get out of control very easily unless it's addressed properly. And the last one there is deterioration in farm infrastructure. So doors, seals, and cracks in floors. We'll describe better as we go exactly how they're implicated. And next slide. So this is a real trick for the modern mushroom farm. On farms that are going through a period of no pest or disease or very little, I always think it's a danger sign because creeping variance happens. Some of the attention to detail into some of the tasks doesn't happen, and then that favours the disease or the pest, and then it lets the enemy get hold. Next slide. And as a final slide for this, I just wanted to say success is like a wheelbarrow with success in hygiene is like a wheelbarrow. If you don't push it, it stops. That's the end of my presentation. So I think
Thank you, Judy. Yep. So I must apologise, Judy. I've inadvertently turned on the timer, so my slides went through on a set time. So apologies for that. I wasn't rushing you at all.
Were you?
Not at all. I wouldn't dare. So thanks for that, Judy. So I'm going to take over now and sort of drill deeper into what Judy has introduced and how these factors of various factors and features help us or hinder us in our efforts to keep a nice clean room for our new crop. So we're going to start with looking at who is the enemy. So what are we up against? Judy mentioned that these were fighting words, and that's not far from the truth because there are many things out there that are determined to damage your business. So in a pest risk analysis about 10 years ago, 15 years ago now, the authors found that crop losses were due to various organisms. And they discovered 20 insect pests, 15 mite species, 11 mycophagous nematode species. They are nematodes that feed directly on the mushroom mycelium and cause damage and loss. And they found 8 saprophytic nematode species. And they are nematodes that live off the bacteria and the solutions in the compost, but do not directly feed on the mushrooms. They have another effect which we'll talk about shortly. They also documented 57 species of weed mold. So we are quite conversant with trichoderma and penicillium. Also things that come across from phase 2, which may indicate problems we have in phase 2. And we can use these to determine what's gone wrong with their crop. And we call those indicator molds. They still cause issues in the crop. But what I want to focus on now are the pathogens that these authors discovered. So these are direct pathogens on mushrooms, and they're classified into 3 groups: the fungi, bacteria, and viruses. So we'll just look at some biology of these things and illustrate why they are so difficult for us to run our mushroom farms in terms of hygiene. So we'll start with the fungi. They're the biggest things, although they start very small. So we have a spore. So that's the infective entity, I guess, of a fungus. It's the most common thing that starts infections. This is a cobweb spore. It's 2-celled, and it produces germ tubes from each end. So there's a bit of growth from each end. The growth is called a hypha. So it's just a thread of cells end on end. And the individual cell is called a hyphal cell. Now, the hyphal cell itself is also able to cause infection because that can germinate in the right conditions. We can't see these hyphae when they're alone or in small numbers, but when they congregate and grow together, we see them as mycelium. So this is the mycelium of Agaricus. Nice, white, rich, vigorous growth. And all that white growth is just a lot of hyphae growing together in such a way that you can actually see it. So the fungal diseases we deal with constantly are, of course, dry bubble, cobweb, and the trichoderma green mold of casing and compost. We refer to dry bubble and cobweb commonly throughout this series because these are the common fungal diseases that we see often, and they also have a different biology. So they need a different management strategy. And we use these as examples throughout. Bacteria are much smaller than our fungus. They consist of a single cell. They have a form of movement, so that a flagellum will just rotate and push the bacterium along in a film of water. Of course, mushroom farms are very wet, and there's a lot of water around, and often the pathogenic bacteria are flagellate, so they can swim around in the film of water. They often have fimbria. So these are little rods that come out of the cell, and these are used for attachment to surfaces, such as mushroom cells, walls, floors, tools, all sorts of things. They're also used to exchange genetic material between compatible bacterial strains. So they have a dual function. Bacteria reproduce by fission. So one cell will split into 2, and each generation will double in size. So approximately a generation for a bacterium is a matter of hours. So, and we're looking at, for an example, the generation time of people is, say, 25 years. The generation time being very short means that there's a lot of mutation in the population, and this is the driver of chemical resistance development. So the shorter the generation time, the more mutations get into the population, and the more susceptible the population is for resistance development. The diseases caused by bacteria, commonly, which we see in Australia, are coarse blotch, various blotches, ginger blotch. There are a number of different pseudomonids now that have been recognised as causing blotch. So the term blotch is not just solely referring to Pseudomonas thalassii and gingeri, also Constantini, Salmonini, and all sorts of other different bacteria that are being identified. The soft rots, we haven't seen any in Australia yet. This is a New Zealand soft rot, so we know it's pretty close. Soft rots will express when the mushroom growing environment overheats, basically. So if your air handling units fail, and your temperature rises, you may have soft rot issues. These are very obvious and will reduce your crop to soup in about 24 hours. So the control measures are pretty simple. Internal stipe necrosis, which we see, the cause of the issue is still debatable. We know that Warwick americana is usually isolated in association with the necrotic stipes, or the dead tissue that forms in the middle of the stipes, but there's no evidence to suggest that the bacterium causes that issue. It seems to be that the issue is a disorder in water relations, and that relationship between the water, the compost casing, and the mushroom evaporation helps the Warwick americana to grow. So it's rather an association rather than a causative relationship. Another bacterium which we find in Australia now and again, or we have in the past, is Warwick Gill, Pseudomonas agaricus. It's another flagellate pseudomonad, so it swims in water and expresses as ooze issuing from the gills and split stipes. It's becoming less common, although the bacterium is re-emerging with different symptoms and has now become another blotch organism. It's very prevalent in Europe, causing issues in Europe. So there are general bacterial diseases which we're looking for. Moving on to viruses. This is a photograph of the France disease. This is the fluid taken out of a diseased mushroom. This was on a photograph taken in 1962, and it was one of the first photographs ever taken by electron microscopy. So that was a new invention back then. And the researcher found that there were two types of virus particles in the juice of mushrooms suffering from La France disease. He found a La France isometric virus, LIV, and a mushroom bacilliform virus, MBV. Interestingly, that it's quite well known now that for a virus disease to express, there must be at least two virus particles present for disease to be expressed. Excuse me. That's the same for mushroom virus X syndrome, as well as for the France disease. So what is a virus? It's a non-living thing, so we can't call it an organism. There's no respiration or metabolism as such. It consists of a short strand of genetic material and a protein coat. It doesn't move in any way. It has no flagella, so it can't swim. Obviously, it has no legs or wings to fly or walk. So it relies on the host to move around. There are two methods of transmission. Horizontal virus transmission, which means that the virus transmits within the crop, within the bed of the same generation. So an infected hypha will approach a clean hypha of the same strain. They will contact, the cell wall will dissolve, and will reform an anastomosis. So an anastomosis is just a joining and uniting of two different septi, two different hyphae, sorry, dissolution of septi. So the virus particles are pushed along by the movement of the cytoplasm or the cell contents within the hypha, and they will pass into the clean hypha and along to infect other hyphae. So this process of anastomosis is just the fusion of compatible hyphae, and that's determined by genetics and chemical signals. And it ensures that the virus cannot spread from genus to genus. It must remain within the same species or even the same variety. It's a very specific interaction. We don't know why hyphae become infected or how they become infected, because unlike viruses such as COVID, these mushroom viruses cannot survive outside the host. So they must remain inside the cells, and as long as the mushroom cell remains viable, the virus will continue to survive. The second form of transmission is vertical. So this goes from one generation to the next generation. Virus particles will pass up into the mushroom cap through the hyphae, into the gill tissue, through the basidium, and into the spore. So as you see here, this is Agaricus bisporus. We have a fat basidium, which gives rise to 2 spores. The virus particles will pass up into the spore, and the spore becomes the main transmission vehicle for mushroom viruses. So that's vertical, going from one generation, and this makes the next generation. And as Judy's explained, we have a mushroom virus X syndrome around. It's characterized by patch disease and also a brown cap mushroom disease, bit of patch, bit of browning. And we also get distorted mushrooms, which are similar to LaFrance disease. So we have dropped veils, tilted caps, and watery streaks on the stipes. These things vary in size. So to put them in scale, this is why we can't see these things. Fungal cell is somewhere between a white blood cell and a grain of pollen in size. A mushroom spore, in which viruses are spread, is somewhere between a red blood cell and a respiratory droplet, so a cough droplet. Bacteria is further down the scale, and our virus is way down the bottom there, and you'll see that it's 0.025 microns, and the micron being 1/1,000,000th of a meter. So the France virus, the LIV, which I showed in a previous photograph, is very, very small, 0.0000025 millimeters. That's why we can't see these things. Right, moving on to the pests. The common pests are nematodes, mites, and flies. The nematodes, as I said before, are either mycophagous, they eat mushroom mycelium directly, or they are saprophagous, so they live on the detritus and the soils and the bacteria and the solutions in the compost. Due to modern composting techniques, we have fewer and fewer mycophagous nematodes around. The major infection is caused by saprophytic Cenorhabditis species, which you have had some experience with recently. While they don't actually directly feed on the mycelium, they do, with their waste products, render the bed uninhabitable by mushrooms, and in fact uninhabitable by themselves. So rather than direct damage on the mushroom, they actually spoil the environment and prevent mushrooms growing. Mites, the red pepper mite, for example, currently Sideroptes, but although it may change its name, as things often do, doesn't directly affect the mushroom, but it makes them look very unappealing and, of course, unsaleable. And as Judy mentioned, they are often in association with trichoderma, primarily, and also other weed molds such as penicillium, Ectomium, and other things that often come through, or not often, but occasionally come through from phase 2. Flies, of course, we're all familiar with flies. This one's a scarab, as we can tell by the V-shaped venation in the wings and the long, thick, chunky antenna. This one's a male because it has a clasper at the rear end. And you can see a dollop of probably Lechnocillium spores on its legs. The flies do directly damage mushrooms, but not in a great deal. You see here that the larvae do cause some issues with mushrooms. The main issue is that the flies will carry diseases, and they are very good at spreading disease such as dry bubble. And mentioning dry bubble and flies, will come to the next section, being vectors. If left alone, diseases basically, fungal diseases like this cobweb will basically remain in place. If you have flies, mites, and other things people going into a room that's heavily contaminated with cobweb, the spores will be disturbed and they'll be spread around the farm. So if left alone, they're not really an issue, but if they are disturbed by something, then they can spread around the farm and cause issues. The things that spread the diseases we refer to as vectors, and that's officially an insect or animal that carries a disease from one animal or plant to another, such as mosquitoes in the human context. Again, referring to our reference fungi, dry bubble and cobweb, they are quite common in mushroom cultivation, but they are different in their reproductive strategy. So dry bubble has a spore that occurs in clumps, and the clump of spores is held together by a sticky mucoid polysaccharide. So that's a sugar, and it's not sticky, thick, viscous, and it holds the spores together. On the other hand, cobweb, the spores are single and they are dry and relatively large. So they can get blown around and they don't stick to anything. Because these are sticky, anything that touches this clump of spores will take that clump of spores to another location, and the spores will stick to whatever they touch subsequently. So a gloved finger will pick up a clump of spores, will touch a wall or a door handle and transfer spores. They will then be picked up by the next person to touch that door or wall, door handle or wall, and so far, and so on and so on and so on. So the spores are spread around. Whereas for cobweb, once they are dislodged, it's more of a airborne problem. So just a reminder of our spores and a hyphal cell, which are both infective. So anytime a spore or a hyphal cell hits the floor or becomes part of the dust, that's available to start a new infection. So these both are infective. If a casing is disturbed which contains dry bubble, then the casing that ends up on the floor and mixed up will break up the hyphal cells, and they will be available to start new infections. So looking at some of the vectors of these diseases, we'll firstly look at the biotic vectors. So that's living vectors. Starting off with our nematodes. So this is our Caenorhabditis, our saprophytic nematode. Excuse me. These are about 1 millimeter or less long. They do not move very far from their range. So any dispersal that they carry out is very limited, very localized. They may churn up the casing and mix in spores with the casing, or they may churn up the casing and expel spores onto the surface of the casing. So they're just very localized, and the spread of fungal material, spores, hyphal cells by nematodes is pretty limited. Similarly, mites, being very small, they have little legs and they don't go very far. The larger mites may be more of an issue because they do in fact travel from room to room. But the red pepper mites have a very limited range. So their impact is minimal. However, there is one species that we know of in Australia that does actually harvest trichoderma. So as Judy said, mites are often found in association with green mold. And this particular species of mite has structures on its underside where it keeps trichoderma spores. So it actively harvests trichoderma spores, tucks them into the little pouches, walks off to a clean bed or a clean area of the bed, plants those spores and produces trichoderma colonies for subsequent generations of this mite. So this mite actively harvests and cultivates trichoderma. We can see where mites go on petri dishes. These are the footprints of mites. So we know they spread bacteria quite well, thank you. Each one of those spots is from a foot or foots. I assume they are feet. And they're very, very good inoculators of bacteria. Now, flies, of course, are an issue that we're all aware of, but why are they such an issue? Again, it's their biology that causes the issues. We have 3 flies here walking over a dry bubble dressing on mushroom. Flies have quite hairy legs, and these are lectinocillium spores or dry bubble spores tangled up in these large hairs on the hind leg. We also see that the lectinocillium spores can fit nicely into the kneecap, and they stick to the leg itself rather than being tied up with the hairs. So the sticky nature of lectinocillium helps to attach to flies, as does the fly hairiness, I guess. So these are scarids, and they're pretty much engineered for fly dispersal, spore dispersal, sorry. This is an indicator of how dirty a fly could be, as Judy showed before from the cobweb farm. This is a puta filled with agar, and one fly was sucked into the puta and allowed to walk over the surface. And we see that there are 1, 2, 3, 4 lectinocillium colonies. So this is dry bubble, a lot of penicillium, and some trichoderma, also a nasty yeast, all off one fly's foot. So you can imagine if you have a room full of flies, how many points of inoculum you're going to have in your room. In addition to vectoring fungi, flies can also move invertebrates around, so our mites and nematodes. We see here a forehead with 1, 2, 3, 4, 5, maybe 6 mites, maybe 7 mites attached to it. So mites are phoretic. They require a vector to move them around the farm. Because they're very small, their range is reduced, and they cannot spread and make new infections without being helped. So when their food is exhausted or their population reaches too high a number, they will start to disperse, and they climb to a high part of the mushroom bed, whether it's a high part of casing or a top of the mushroom, as we see swarming mushrooms before. They will stand on their hind legs and put their forelegs in the air, ready to catch a fly as it goes past. Then they climb on its back and get carried off to a new room or a new bed, and they'll start an infestation elsewhere in new pastures. Similarly, the nematodes also, when their environment is fouled so much that they have to move out, they will enter a growing stage called a dewar larva, where the metabolism slows down. They will climb to a high part of the casing. They will congregate into groups of 200 or 300, and they'll start waving at flies. And it's often termed winking. So they attract flies by movement. Flies come along to investigate, and the nematodes will attach to the flies and get carried off to new parts of the mushroom farm to establish a new infection. Now, these things are also affected by human activity. And by human activity, we mean the normal processes that go on day to day in a mushroom farm, the standard operating procedures of a mushroom farm. We've all got some of these on farm. The mushroom harvester. Are they devils or angels? The issue with dry bubble, again, as I've mentioned before, is that the spores are sticky and they occur in clumps in a high number of spores and clumps which stick to anything. So if a harvester touches a mushroom expressing dry bubble, they will pick up the spores and transfer them wherever they're going next. To demonstrate how difficult this is, we ran a test at the Marsh Lawson Mushroom Research Unit a number of years ago in a previous project, and we inoculated a crop late with lectinocillium. We see it here as expressing with browning, and the grey velvety growth is sporulating lectinocillium. So Judy touched her fingers onto a patch of sporulating Echinocilium and touched her fingers onto a plate which was formulated to grow Echinocilium selectively. And we got pretty much pure Echinocilium directly off. In the second experiment, she took one finger, one touch onto a mushroom. Then she was able to touch 96 times onto a petri dish, and we're still getting growth after 96 touches off that one initial touch. This demonstration was done previously by John Fletcher, who got up to about 400 touches before the supply on his finger was exhausted. So a single touch can spread these sticky spores a long, long way. So the issue for harvesters is if we have nice, clean mushrooms, these can be hiding the bubble. So we've got 1, 2, 3, 4, 5 bubble there. And if a harvester is speeding along, trying to get their quota up, we find that often they will pick up bubble inadvertently, and we call these incidental touches. Similarly, some nice mushrooms here, but hiding bubble. So it's just an inadvertent touch on a crop that has not been made safe previously before harvesting. Another issue with the spreading of dry bubble is ineffective spot treatment. So spot treatment, we know, is a long, tedious, quite boring business, but it must be done correctly. And if it's not done correctly, it results in spreading the disease rather than containing the disease. So often the result of harvesting and spot treatment is a pile of casing and infected mushrooms lying on the floor. As these dry out, the spores and hyphal cells of the pathogens become part of the room dust. And once the brooms are brought out for sweeping, that dust is aerosolized, and these spores and infective cells fly up into the air and they get blown by air handling units, open doors, and even just people's movements, spread the dust around and therefore vectoring the diseases. Another common cultivation practice is, of course, watering. Overspray of a crop that has not been made safe. So watering on disease will certainly splash the pathogens onto the floor. Once on the floor, the pathogens are often lodged in cracks, and they are also packed in there with organic material such as casing, compost, and mushroom debris. We know for a fact that mushroom floors do not reach kill temperatures during cookout, and if they're not sanitized correctly, they provide significant reservoirs for disease expression. Now, noting that our definition of vectoring was more aligned with living things, I've stretched that definition to abiotic factors that aren't alive. So not small dogs, but wind, construction, recycle boxes, and machinery. So we've touched on construction briefly, but dust, which we'll explain in a further session, is a very big issue on a lot of farms. Excuse me, on many farms. As we can see here on the stored compost, casing, stored peat, there's a lot of dust collecting, and that is guaranteed to contain some pathogen spores and pathogen hyphal cells. But how effective is the dust? Well, Helen Grogan performed an experiment many years ago where she took 4 treatments and applied them to mushroom beds. So she took a high Echinocilium spore load of pure spores at 1 million spores per square metre and applied it to the bed. Similarly, she applied pure Echinocilium in a low load of 1,000 spores per square metre. She took some dust from a grow room floor and applied it similarly, and then dust from a roadway outside grow room and applied it to. She found that on a high load of spores, bubble was expressed in first flush, but by the third flush, the crop was wiped out. On the low concentration of pure Lechinocilium, there was nothing on the first flush. By the third flush, dry bubble was expressing. With the casing grow room dust, similarly, no bubble in the first, but bubble expressing in the third. And for the roadway, there was no bubble expressing until late in the third flush. So she noted that these two treatments, the low concentration of pure Lechinocilium and the grow room dust, had similar infectivity. And we can see from this that the grow room dust is highly significant in the development of mushroom disease, particularly dry bubble. So as Judy alluded to, construction is a high risk exercise on a mushroom farm. Anything that disturbs the environment, even renovations of existing infrastructure or fitting new air handling units, ventilation, anything that disturbs what's already there creates a serious risk of disease forming. Groundwork, even on adjacent properties, is again a high risk factors for mushroom disease. And talking about the floor being the receptacle for most things, through our standard operating procedures, we know that from practice that any wheeled vehicle will pick up these the diseases from the cracks and other reservoirs in the floor and carry them around the farm. So any wheeled vehicles, being picking trolleys, picking stools, forklift trucks, pallet trucks, any wheeled vehicle is very likely to pick up spores on the floor and drag them around the farm. So the next quick topic we'd like to cover is on-farm IPDM. So Judy mentioned that IPDM is composed of a number of events, of which hygiene is pretty major. IPDM, often referred to just as IPM, where pest is referring to anything that causes an issue, I guess. It's a system of complementary technical and management methods which suppress or eliminate the pathogens in a safe, cost-effective, environmentally sound manner. So the advantage is that we are reducing reliance on expensive and highly toxic synthetic pesticides. And we're looking at replacing these with less impactful, I guess, biological alternatives. It also reduces the impact of synthetic pesticides on the environment. So, for example, fipronil we know can be used on farm, but there is an issue with bees. And in fact, fipronil is under review at the moment by the APVMA. So an effective IPDM program also reduces the effects of synthetic pesticides on staff. So wherever you have a nasty chemical, a toxic pesticide, staff contact is always going to be an issue. And IPDM programs are likely to reduce the chance of resistance developing in the pathogens. Now, the main aim of the IPDM is that it's going to reduce the number or the population of pathogens on the farm to a safe level that is below the threshold of disease. So we're not trying to sterilize the farm. We're not trying to eradicate everything, because that is impractical and it's never going to happen. We just need to reduce the pathogens below the disease threshold, at the level at which disease expresses. So there are 7 key elements to an effective mushroom farm IPDM program. We'll often use the word effective because anyone can start a mushroom farm IPDM program, but it's no good starting one if it's not going to be effective. So we always qualify IPDM as being an effective program. So first step, and these steps are not necessarily in this order, depending on your farm, you must identify the key pests and pathogens. So therefore, once that's done, we can develop specific controls or containment measures. Knowing what we're dealing with will also narrow down the management options. So we know there are a few products available on the APVMA if we choose to go that way. And this will narrow down what we can use and what we can consider, knowing what we're dealing with. And of course, knowing what we have to deal with, we can actually choose the correct biological product and not necessarily resorting to the high toxic chemicals all the time. So we need to know the enemy. So it's not just about knowing what we have, but how it lives, how it reproduces, where it infects on the farm, when it infects on the farm. And by doing that, we can not only target the organism, but we can target the time it infects and the location where it infects. And we can identify where on the farm we have to concentrate our actions. There's some areas of the farm may be more vulnerable than others. Determine the thresholds, which simply means that at what stage do we consider this pest or disease economically damaging? When do we start throwing money at this problem? Can we withstand some loss of product before we spend a lot of money on managing this thing? Also, we need to determine the disease threshold. How much of the pest or pathogen can be present before we start seeing a loss of crop, a loss of quality, a loss of income? And that's also connected to the economic threshold. At what stage do we start spending money? A major element of an effective IPDM program is that we can apply preventive cultural practices. So we're favouring the mushroom and disfavouring the pest or pathogen. And by knowing what pathogen we have, we can perhaps tickle our cultivation accordingly. But it's not negotiable that we must provide a highly selective compost in the first instance. So that not only promotes vigorous growth from the mushroom, but it also dissuades our pests and pathogens and prevents them colonising compost and casing. So we need to ensure the environment favours agaricus. We have to check that our pig heat is certainly effective, and we must ensure that our stringent whole farm hygiene program is in place and is operating well. We must also develop and implement an effective management strategy. So looking at all our physical controls, our biological controls where available, and chemical controls if necessary. So we're not saying don't use chemicals, we're trying to reduce their use because they are a big hammer, they are effective, and they are expensive. And there are alternatives. By reducing the pathogens on the bed or in the mushroom house, we can reduce the reliance on the chemical controls. Monitor, monitor, monitor. This will help us detect new infections, new pests and pathogens, and it will also give us a heads up on when things are increasing, like flies, for example. Increasing fly populations suggests that we may get an issue with diseases, particularly dry bubble, in the very near future. And we can also use monitoring to check that our hygiene program is effective or not. Now, we started out by saying there are 7 elements. We've only got to number 6. The IPDM, to be effective, must be a continual process. So we're looking at an IPDM continuum. So we'll have our 6 features, our 6 strategies. But the most important one is number 7, evaluate and follow up. IPDM cannot be effective if it is just a linear process. You start at A and stop at C or let it peter out. You must evaluate and follow up by reviewing your monitoring data and ask yourself if your response is effective And if not, why not? Is the timing appropriate? If not, why not? For example, are you treating for flies, adult flies, at the correct time? Or are you trying to treat larvae at the correct time? Don't be afraid to consult your supervisors and pickers, because they are at the front line. Also, be prepared to adapt your response and implement your modified response. And again, monitor, evaluate, monitor, evaluate. That's probably the key step to ensuring your IPDM is effective. Always reassess. Now, looking at some activities on the farm which we do, I guess, naturally, but are in fact elements of an IPDM system, an effective IPDM system. IPDM classically is described as exclusion, containment, and elimination in those three categories. But because we are growing in an enclosed environment, often our exclusion practices are also our containment practices. So let's have a quick look through these. Exclusion. We know that we've got to get rid of our vectors. We've got to keep vectors out of our clean room because they'll bring in dirty things from other rooms. So we're looking at door seals, fly mesh, strategic sticky fly papers, foot dips, signage, and traffic control. So the last two are more for our human vectors. So the last three, in fact, more for our human vectors, because we want to keep their feet clean, because we know the floor is a huge reservoir. We want signage to warn people not to go in a room, and we need traffic control to make sure that our third flush harvesters don't go back to first flush on the same day without changing their clothes first. Dust control. We don't want dust containing all our particles, all our spores and spores and fungal cells to hit the bed. So we're making sure our door seals are tight and they are in good condition, and also check that the door is hanging correctly. A new door seal doesn't do much if the door is not hanging properly. And make sure your air is filtrated, or filtered, I think is the word. Spot treatment, disease monitoring and recording. While it's not a exclusion practice, it was certainly it's very useful to know when to start our exclusion or when to reassess our exclusion and make sure everything's hunky-dory. Pest monitoring and recording, similarly, after training. These are the people that spot disease more often than not. So they need to recognise the symptoms and know how to report symptoms and how to isolate what they see. Strict gov policy will prevent nasties getting into a new room. And again, signage to make sure no one gets into a diseased room or where they're not supposed to go. In terms of elimination, we can start by making sure that pests and diseases are unable to grow in our mushroom compost by making sure it's highly selective. Our cultural practices must be top-notch, so our environment control, make sure our handling units are calibrated and running well, the filters are clean, and the airflow is as expected. Spot treatment, although this is a containment, it does to some degree eliminate the issue. Fly control, we can kill adult flies with electronic zappers to a certain degree by strategic sticky yellow fly traps, but we can also use pesticides, insecticides. We can look at our fungicides as required and particularly make sure our waste disposal protocol is effective and efficient. So make sure that your spent compost is taken away off the farm as soon as possible, and your excess phase 3 compost is not dumped on the farm. That is a virus hazard. So looking over to our whole farm hygiene, we'll be looking at this in particular next week. So grow room hygiene, the basic tenets there are make sure your cookout is working, so calibrate your probes, making sure you're getting up to temperature for the right time. Keep your organic material out by cleaning with detergents, soap and water, then followed by disinfection or sanitation. Foot dips well maintained and monitored for organic material. As we'll see shortly, that organic material does affect foot dips severely, and it doesn't take much to do that. Monitoring, self-explanatory. We've been through monitoring and what it's good for. Sanitation of tools, sanitation of machinery and facilities. So we're trying to reduce the number of pathogens and pests on our farm, on our tools, machinery and facilities to below the threshold level for disease. Contact services such as maybe benches, tables where spores could land, particularly Lechinocilium could land and be stuck so that they are picked up by subsequent people coming along. We need to make sure our processes are scheduled so we don't obviously fill a room while we're emptying another room at the same time. Make sure our strict glove policy is in place and is enforced. And please make sure your clean clothing is a daily occurrence and not just a weekly occurrence. So that's my presentation for this introductory session done. Please feel free to contact either Judy or myself for follow-up questions if you have. And next week, we're looking at effective cleaning, choosing detergents and disinfectants, the effect of detergents, influence of organic material and disinfectants, economic thresholds, and a specific case study. I think I'll hand you back to Leah.
Thank you, Wal, and thank you, Judy, for this presentation. I always love listening to the two of you speak. I always learn something, which I just think is just so fascinating. I had no idea that mites could attach to flies, or that there was a mite that was harvesting trichoderma and transporting it to other parts. And I think that's just really fascinating. Some of the images that you showed were like scenes out of a horror movie, weren't they? And they certainly illustrates why integrated pest and disease management is so important, starting with good farm hygiene as a preventative measure, but also effective staff training, why that's so important as well. Unfortunately, we've run out of time for Q&A today, but I do encourage you, if you do have any questions regarding this webinar or anything specific to your farm, that you reach out to Judy or Wal directly. They would be happy to answer any questions that you have. Before we wrap up today, I'd like to ask each of you to please stay online after I terminate, or after we finish this webinar. The screen will bounce to a digital survey. It's really important that you take the few minutes. It's only 3 questions. It'll take you 2 minutes to do it, but it's imperative that the project team gather your feedback to aid future funding for educational programs to support the industry. So that concludes today's AMGA webinar. Thank you to all who attended. Thank you again to Judy and Wal. As previously mentioned, a recording will be sent to all who registered to attend today, as well as being housed on Agora, our industry-funded pest and disease information library. Thank you all very much and good afternoon.
Thank you.